My name is Nila Kamal, and I am from Sharjah English School. Today, my presentation will be investigating serotonin receptors in the age of mental health crises. A little about myself. I am 16 and a rising junior. I'm also an aspiring neuro-oncologist who is interested in choir, public speaking, debate, and acting. I've previously done some research at the Science Internship Program at UC Santa Cruz. Now, why should you care about the research? For every young adult watching this video, how many do you think suffer symptoms of mental illness? 1 in 30? Well, maybe 1 in 10. 1 in 7 adolescents around the world suffer from a mental health condition, yet many of them will never receive adequate treatment, making mental health one of the biggest threats to achieving global health. When global disparities come into the picture, stigma is one of them. A global survey across 8 countries found that 74% of people said that stigma was the primary reason that they would not seek treatment. In 58 low middle-income countries, the darker areas show places where there are far too few mental health professionals to give anyone proper care. And issues with current medication are expensive treatment. Antidepressants like Cymbalta can cost up to $2,000 per month. And of course, treatment resistance. 70% of people suffering from depression respond well to therapeutic drugs, but 30% are treatment resistant. We urgently need new science-driven solutions that reach everyone, especially that 30%, in order to achieve global health. And here's where my research comes in place. The use of psychoplastogens in healthcare, an opportunity for scalable and more affordable treatment with longer-lasting effects. Psychoplastogens are a group of small molecule drugs that produce effects on the neuronal structure and function. They are known to have therapeutic and hallucinogenic effects. Clinical studies showed that psychoplastogen can significantly reduce symptoms of depression, sometimes with effects lasting for months. Patients report improved moods and emotional stability. In clinical trials, 67% of patients with treatment-resistant depression responded well after psilocybin therapy, a type of psychoplastogen. And of course, there are some hallucinogenic effects, which depend solely on the dosage and a person's mental state prior. This involves visual and auditory distortions. Now, following this, we decided to study these effects using mice as human proxy. On a side note, I will be focusing on the therapeutic effects of psychoplastogens more than the other. One of the ways that scientists have been able to test the potential for therapeutic benefit of these psychoplastogens is through the formation of dendritic spines. Experiments on depressed mice treated with psychoplastogens found correlation between neuronal spine growth and their behavioral responses, leading them to believe that spine formation can be used to measure therapeutic potential. Healthy dendrites have a lot of spines, but mental illness reduces those spines, which severely affects the neuron's ability to function properly. When dendrites are treated with these compounds, there is a notable increase in spine formation, alluding to the potential for the therapeutic effects. Following this, we decided to focus on a serotonin receptor subtype known as a 5-HT2AR receptor. This image shows the key serotonin receptors in the brain and also shows the exact target site for many antidepressants. The receptor itself plays a vital role in perception and affecting moods. In our experiment, we worked with 2 types of mice. The wild-type mice, which are normal mice with no genetic changes, and they serve as our baseline. Then we had the knockout mice, which were genetically modified so they don't have the 5-HT2AR receptor. Now for the methods. We wanted to know, is the receptor necessary for psychoplastogens to work? We then started to form our hypothesis. If psychoplastogens rely on this receptor to work, then the knockout mice shouldn't show any healing effects. We first perfused the mice and collected the brains. The brains were sliced on this machine called the vibratome, which slices the brain into thin slices that are approximately 40 microns thick, so light can pass through them as we image them. We then stain the slices with immunohistochemistry, which uses antibodies and fluorescence to mark where certain receptors are. The primary binds to the 5-HT2AR, which binds to secondary. The third was coupled with a fluorescent molecule, which binds to the secondary. We also counterstain the slices with DAPI, a fluorescent stain that binds strongly to certain proteins in the nucleus of a cell. This marks where each cell was to help us find our region of interest when analyzing. During the immunostaining process, we also analyzed the images of brain slices on Fiji, and our region of interest was the primary somatosensory cortex of the brain, which plays a vital role in using motor skills for skilled movement. This is the DAPI region, and this is a red channel where we did most of our research. When looking at the wild-type mouse, we saw a dense, bright band in that region. This strong fluorescence tells us that the 5-HT2AR receptor are highly expressed there. In contrast, the knockout mice showed little to no brightness in that region, given that they don't have the 5-HT2AR receptor. The peak is at 200 microns, whereas in knockout, there is no peak, which ultimately supports our hypothesis. Without the receptor, psychoplastogens offer no therapeutic benefit. For the future, we decided on some future experiments. The conditional knockout of the 5-HT2AR receptor, meaning the receptor would be expressed everywhere except the neurons we're focused on. It would help us test out the therapeutic effects that are unique to those specific brain cells, or it would also help us find out if other areas of the brain play a role in the activation of the psychoplastogens. The observation of such effects produced by psychedelics are among the most recent exciting discoveries in modern psychiatry. It is important that the scientific community optimistically reopen the doors to the possible clinical use of these compounds. Thank you to my mentor, Stefan, and the Science Internship Program, and of course, to my friend, Kaylin. Thank you for listening. For any further doubts or clarifications, my email and LinkedIn is attached.